Hello students, I am Dr. Nimi Singh. Today I bring you the next learning episode in BSc Forensic Science on a very important unit of paper, Technological Methods in Forensic Science, that is Forensic Microscopy. This particular topic will be dealt in two parts today in this lecture, that is Forensic Microscopy Part 1. You will be introduced to the concept of microscopy. You will also be acquainted with the principle and the components of a microscope. You will also learn about the different types of microscope. Lastly, we will wind up this module by understanding the applications of microscopy in forensic science and conclusion. Dear students, let us have a look at the modules which we are going to study today. The first one was going to be Introduction to Microscopy, then Principle of Microscopy, Components of a microscope, types of microscopes, applications of microscopy in forensic science, and then in the end the conclusion. Let us come to the first topic, that is the Introduction to Microscopy. A microscope is well defined as an optical apparatus that uses an arrangement of lenses to yield magnifying image of small objects. To achieve this, a stereo microscope uses a number of mechanisms that gather light and then transmit the light track so that a magnified image of the observed object can be focused within a very short distance. The discipline of examining small things using such an instrument is what is called as a microscopy. Microscopic means undetectable to the naked eye if not assisted by a microscope. There are numerous types of microscopes, and optical microscope is the most up-to-date and the first manufactured microscope which utilizes light to image the sample. Electron microscopes are another category of microscopes which comprise of transmission electron microscope and scanning electron microscope. The first microscope to be technologically advanced was the optical microscope, though the original is not easy to recognize. The primary microscope was believed to be made in 1590. Giovanni Faber coined the word microscope for Galileo's compound microscope in 1625. The optical microscope, time and again referred to as a light microscope, is a kind of microscope which involves visible light and a coordination of lenses to enlarge images of small samples. Optical microscopes are the elementary design of a microscope and were designed around 1600. Elementary optical microscopes can be very simple, although they are rare, countless, multifaceted designs which intends to have advanced resolution and sample contrast. Factually, optical microscopes were easy to improve and are prevalent because they use visible light, and as a result the sample can be easily observed by naked eye. The image from an optical microscope can be taken by standard light sensitive cameras to produce a micrograph. Initially, images were taken by photographic film, but contemporary improvements in CMOS and later charge coupled device cameras permit the storing of digital images. Only digital microscopes are now accessible, in which a CCD camera is utilized to analyze a sample and the image is then displayed directly on a computer screen devoid of the requirement for eyepieces. A substitute to optical microscopy which does not apply visible light comprises of scanning electron microscopy and transmission electron microscopy. There are two rudimentary arrangements of the conventional optical microscope, the simple with one lens and compound with many lenses. The immense preponderance of present study microscopes are compound microscopes, while some economical commercial digital microscopes are simple single lens microscopes. Principally, a magnifying glass is a rudimentary single lens microscope. In general, microscope optics are stagnant to concentrate at diverse focal depths. The lens to sample distance is adjusted and to receive a wider or narrower field of view. a separate magnification objective lens should be applied. Most up-to-date research microscopes also have a separate set of optics for illuminating the sample. Let us now come to the principle of microscopy. A usual microscope comprises of an objective lens, ocular lens, lens tube, stage and a reflector. The objective lens magnifies the specimen placed on the stage. The magnified image of the specimen, when focused, can be observed through the ocular lens. A microscope is designed to emit light onto or through objects and magnify the transmitted or the reflected light with objective and ocular lenses. Coming to the components of a microscope. All contemporary optical microscopes aimed for observing samples by transmitted light have the similar uncomplicated apparatuses of the light direction, listed here in the order in which the light travels through them. The source of light, a light or a mirror, diaphragm and condenser lens, objective, ocular lens. In addition, the vast majority of microscopes have the same structural components: objective, turret, stage to hold the sample, focus wheel to operate the stage. Ocular lens, that is the eyepiece. The ocular lens or the eyepiece is a cylinder comprehending multiple lenses. Its purpose is to converge the image in focus. The top of the body tube has an eyepiece inserted into it. These eyepieces can be switched with numerous other eyepieces which can be inserted with variable degrees of magnification. Magnification values for eyepieces comprise of 2x, 5x and 10x. Matching of optical configuration of objective lens and eyepiece is done in few high performance microscopes to give optimum optical performance. This happens ordinarily with apochromatic objectives. Now, what is a stage? A podium which holds the specimen which is being viewed is known as stage. It is present beneath the objective. A hole is present in the center of the stage to let the light pass for the illumination of specimen. Arms are usually present to carry the slides. Now, the different light sources. Various sources of light can be used. At its simplest, daylight is directed via mirror. Most microscopes nevertheless have their own manageable source of light, generally a halogen lamp. Now, coming to condenser. The condenser is a lens designed to focus light from the illumination source onto the sample. The condenser may also include other features such as diaphragm and or filters to manage the quality and intensity of the illumination. For illumination methods such as dark field, phase contrast and differential interference contrast, microscopy and additional optical components should be precisely aligned in the light path. Now, what is a frame? The complete optical arrangement is conventionally fixed to a rigid arm, which is further joined to a strong U-shaped foot which provides the required rigidity. The viewing angle can be adjusted using an arm angle. The frame provides a mounting point for various microscope controls. Various panels for focusing like large knurled wheel to discuss coarse focus, smaller knurled wheel to regulate fine focus are present in the frame. The controls for lamp and condenser are some other features which are mounted on the frame. Now, what are objective lenses? On a typical compound optical microscope, there is a selection of lenses available for different applications. Many different objective lenses with different properties and magnification are available. There will be around 3 objective lenses: a low power lens for scanning the sample, a medium power lens for normal observation and a high power lens for detailed observation. The typical magnification of objective lens depends on the intended application. Normal groups of lens magnifications may be 4x, 10x, 20x for low magnification work and 10x, 40x, 100x for high magnification work. Now, what is a magnification? The original power of a compound optical microscope is the outcome of the powers of the ocular and the objective lens. The maximum normal magnifications of the ocular are 10x and objective is 100x, which gives an ultimate magnification of 1000x. Now, let us come to the types of microscopes. There are various types of microscopes available. It depends on the type of microscope which can interact with the sample readily to produce an image. You have single lens, that is a simple microscope. The simple microscope, being the original version of light microscope, comprises of a single lens only for the purpose of magnification. Anton van Leeuwenhoek's microscope consisted of a single and simple convex lens attached with a plate of brass and having a screw mechanism so as to hold the sample or the specimen that was to be examined. Demonstrations by British microscopists have images from such basic instruments. Although it is now considered very basic, but the use of single convex lens for the purpose of viewing can still be found in simple magnification devices, for example, in magnifying glass and the loupe. Then we have the compound microscope. The compound microscope employs multiple lenses so as to collect light from the specimen, and then this light is focused into the eye with a separate set of lenses. In comparison with simple microscopes, the comparison microscope is expensive, large and heavy because of the additional set of lenses used in the construction. The chief benefits of the additional lenses are decrease in chromatic aberration, improved numerical aperture and replaceable objective lenses for modification of magnification. This type of microscope also creates more improved illumination setups, for example, a phase contrast. Fluorescence microscope. Today, the biological microscopy is dependent on the development of fluorescent probes for studying the specific structures within a cell. In comparison to normal trans-illuminated light microscopy, in fluorescence microscope, the sample illumination is carried out by the objective lens with the help of a narrow set of wavelengths of light. The light emitted initially interacts with the fluorophores present in the sample, which then emit light of a longer wavelength. It is this emitted light which makes up the image. Then comes the stereo microscope. The stereo or dissecting microscope is an optical microscope variant designed for low magnification observation or a simple using incident light illumination rather than trans illumination. It uses two different optical paths with two eyepieces and two objectives in order to deliver slightly separate viewing angles to the left and right eyes. A three-dimensional imaging of the sample is produced in this way. The stereo microscope is frequently used to analyze the surfaces of solid specimens or to carry out close work such as sorting, dissection, microsurgery, watchmaking, small circuit board manufacture or inspection, etc. The stereo microscope should not be mixed with a compound microscope fitted with a double eyepiece and a bino viewer. In such microscopes, both eyes get the same image, but the binocular eyepiece deliver greater observing comfort. However, the image in such a microscope is no different from that obtained with a single monocular eyepiece. Now, coming to comparison microscope. A comparison microscope is a tool which is used to examine simultaneous specimens. It comprises of two microscopes linked by an optical bridge, which results in a split view window allowing two separate objects to be viewed consecutively. This prevents the viewer having to depend on the memory when associating two objects under a conventional microscope. The modern apparatus has various optical, mechanical and electronic refinements including fiber optic illumination, video capabilities, digital imaging, automatic exposure for conventional photography, etc. Then you have is the petrographic microscope. A petrographic microscope is a type of optical microscope which is used in petrology and optical mineralogy to identify rocks and minerals in thin sections. The microscope is used in optical mineralogy and petrography, a branch of petrology which focuses on detailed descriptions of rocks. The method is called polarized light microscopy. Depending on the grade of observation required, petrological microscopes are derived from conventional bright field microscopes of similar basic capabilities by adding a polarizer filter to the light path beneath the sample slide, replacing the normal stage with a circular rotating stage typically graduated with vernier scales for reading orientations to be better than 1 degree of arc. Adding a second rotatable and removable polarizer filter called the analyzer to the light path between objective and the eyepiece. Petrographic microscopes are constructed with optical parts that do not add unwanted polarizing effects due to stained glass or polarization by reflection in prisms and mirrors. Coming to the phase contrast microscope. Phase contrast microscopy is an optical microscopy illumination technique in which small phase shifts in the light passing through a transparent specimen is converted into amplitude or contrast changes in the image. A phase contrast microscope does not require staining to view the slide. This type of microscope makes it possible to study the cell cycle. The phase contrast microscope is widely used for examining specimens such as biological tissues. It is a type of light microscope that enhances contrast of transparent and colorless objects by influencing the optical path of light. Then we have is the electron microscope. Electron microscopes are scientific instruments that use a beam of extremely energetic electrons to study objects on a precise scale. This examination can produce information about the topography, morphology, composition and crystallographic information about the object. Light microscopes had certain limitations as the physics of light to 500x or 1000x magnification and a resolution of 0.2 micrometer, instigating thereby the development of electron microscopes. In the early 1930s, this theoretical limit had reached and there was a scientific aspiration to comprehend the fine details of the interior structures of organic cells like nucleus, mitochondria, etc. This required 10,000x and above magnification, which was just not imaginable using light microscopes. So, electron microscopy has been further classified into two types: transmission electron microscope and scanning electron microscope. Here in today's lecture, I will be just giving an overview of these types of microscopes. We will be studying the respective types in detail in our subsequent lecture. The transmission electron microscope was the first type of electron microscope to be developed and designed accurately on the light transmission microscope, except that a focused beam of electrons is used as a replacement for light to see through the specimen. The transmission electron microscope was devised by Max Noll and Ernst Ruska in the year 1931 in Germany. On the other hand, the first scanning electron microscope was found in the year 1942, with the first commercial instruments coming in around 1965. Its delayed development was because of the electronics behind scanning the beam of electrons through the sample. Most microscopes can be classified as one of the three basic types, which are optical, charged particle electron, or an ion or scanning probe. let us come to the applications of microscopy in forensic science. There are limitless applications of microscopy in the field of forensic sciences. The microscope is used to form an enlarged image of a small object. After enlargement of the image, the next concern is the resolution, that is, the ability to see increasingly fine details. The technique of microscopy is used to examine many evidences in forensics, such as ink lines in case of forgeries, blood stains, bullets, etc., and any other examination is not required in many situations. For examinations of such kind of evidences, the evidence may be studied directly under the appropriate microscope without any sample preparation. Based on the investigation required, different types of microscopes are available and used for examinations. These microscopes have paved their way in the field of forensics too. They are being used for analysis and examination of samples. Few applications of various microscopes have been listed here. First is the optical microscopes. They are employed in combination with visible light and transparent lenses, being the most familiar ones because they are used to see objects as small as about 1 micrometer. For example, in case of red blood cells, which are 7 micrometer in size, or a human hair 100 micrometer. The surfaces of solid specimens can be studied under stereo microscope, or they are also used to carry out close work, for instance, in case of sorting, dissection, microsurgery, etc. The phase contrast microscope is generally employed for the examination of specimens such as biological tissues. Phased contrast microscopy is a type of light microscopy which enhances the contrast between the transparent and colorless samples by influencing the optical light path. Cell constituents or bacteria that cannot be perceived by the naked eyes can be easily visualized with the help of a phase contrast microscope. Now, the microscope remains the most important and useful instrument for locating and comparing specimens of paint. There are variety of paint shades and colors, and hence paint evidence serves as the most distinctive form of evidence. The color, the sequence of layers and surface texture in case of paints can be easily studied under the stereo microscope. Also, the stereo microscope can be used for viewing opaque samples and in the characterization of physical evidences such as soil, paint, gunpowder residues and marijuana. The examination and comparison of firearms were refined by Colonel Kelvin Goddard by employing the comparison microscope. The work carried out by Goddard allowed firearm examiners to determine if a particular firearm fired a bullet by the comparison of question bullet with the test fired bullet from the suspect's weapon. Using a compound light microscope with magnification capabilities up to 1.0003, analysts usually can identify pollen and spores as having come from a specific plant family or genus, and sometimes even the unique species. Many of the biological samples such as blood cells, seminal stains and vaginal cells etc. are being examined with the help of compound microscope only. The ballistics identification network, that is the drug fire system, and NIBIN also employs comparison microscope with the purpose of comparing bullets and cartridge cases procured from the scene of crime and those already stored in the databases. Handwriting samples are often examined with the help of stereoscopic microscopes. Let us come to the conclusion of our lecture. Microscope is an indispensable tool in the field of science. It helps in observing the minute details within the sample which are being examined by the help of emitted light onto or through objects and magnifying the transmitted or the reflected light with the objective and ocular or eyepieces, lenses. The magnified view of the object helps in locating the minute peculiarities in them. This property of microscopy has been used in the field of forensic science. Various types of microscopes are being used today with the purpose of analyzing samples that will help in forensic casework investigation. With all these information, here we come to the end of today's lecture. Do keep in mind what we have discussed today. I will be back with one more lecture in this series. If you want to learn more and enhance your knowledge, you may log on to our website www.cec.nic.in for MCQs, quizzes, LORs, etc. Thank you for your time today. I will see you in the next lecture. Have a great day.